In this section, we will talk about depolarization of multi-unit smooth muscle without action potentials. The smooth muscle fibers of multi-unit smooth muscle, for example, the muscle of the iris of the eye, or the piloerector muscle of each hair, normally contract mainly in response to nerve stimuli. The nerve endings secrete acetylcholine in the case of some multi-unit smooth muscles and norepinephrine in the case of others. In both cases, the transmitter substances cause depolarization of the smooth muscle membrane, and this depolarization in turn elicits contraction. Action potentials usually do not develop because the fibers are too small to generate an action potential. When action potentials are elicited in visceral unitary smooth muscle, 30 to 40 smooth muscle fibers must depolarize simultaneously before a self-propagating action potential ensues. However, in small smooth muscle cells, even without an action potential, the local depolarization, called the junctional potential, caused by the nerve transmitter substance, spreads electrotonically over the entire fiber and is all that is necessary to cause muscle contraction. Now, let's discuss. Local tissue factors and hormones can cause smooth muscle contraction without action potentials. Approximately half of all smooth muscle contraction is likely initiated by stimulatory factors acting directly on the smooth muscle contractile machinery and without action potentials. Two types of non-nervous and non-action potential stimulating factors often involved, and that are local tissue chemical factors and various hormones. Now, let's discuss smooth muscle contraction in response to local tissue chemical factors. The smallest of these vessels have little or no nervous supply. Yet, the smooth muscle is highly contractile, responding rapidly to changes in local chemical conditions in the surrounding interstitial fluid and to stretch caused by changes in blood pressure. In the normal resting state, many of these small blood vessels remain contracted. However, when extra blood flow to the tissue is necessary, multiple factors can relax the vessel wall, thus allowing for increased flow. In this way, a powerful local feedback control system controls the blood flow to the local tissue area. Some of the specific control factors are: first is, lack of oxygen in the local tissues causes smooth muscle relaxation and therefore vasodilation. Second is, excess carbon dioxide causes vasodilation. Third is, increased hydrogen ion concentration causes vasodilation. Now, moving forward, adenosine, lactic acid, increased potassium ions, nitric oxide, and increased body temperature can all cause local vasodilation. Decreased blood pressure, by causing decreased stretch of the vascular smooth muscle, also causes these small blood vessels to dilate. Now, moving towards effects of hormones on smooth muscle contraction. Many circulating hormones in the blood affect smooth muscle contraction to some degree, and some have profound effects. Among the more important of these hormones are norepinephrine, epinephrine, angiotensin II, endothelin, vasopressin, oxytocin, serotonin, and histamine. A hormone causes contraction of a smooth muscle when the muscle cell membrane contains hormone-gated excitatory receptors for the respective hormone. Conversely, the hormone causes inhibition if the membrane contains inhibitory receptors for the hormone rather than excitatory receptors. So this was all about this video. Stay tuned to sqadia.com for more precise and knowledge-stuffed videos.
Explore our extensive library of over 1,800 video lectures to learn about a wide range of topics. Only on sqadia.com.